Good morning, Morley, and thanks for being here today. This one's been a long time coming, and I'm glad we found time to finally dig into everything copper and iron today. Now, I was fortunate enough to have a chance to listen to you lecture in London a few months ago. It was an amazing experience, and I learned a lot, especially about copper as it pertains to sort of post-viral fatigues. Of course, I don't want to get ahead of myself, and I know that today's conversation will be aimed maybe more at the basics, and then hopefully we can get together at a later date and dig into sort of more specific topics. Anyway, before we start, would you mind introducing yourself to those in the audience who maybe aren't familiar with you and your body of work?
Sure. Well, thank you for the opportunity, and it's great to meet you, and look forward to our discussion now and maybe going forward. I'm Morley Robbins. I affectionately refer to myself as a pre-med retread. I had designs of going to medical school, and a very good friend of mine, Ben Edwards, who's a physician in Texas, said, "It's a blessing you didn't go to medical school." And I said, "Why is that?" And he said, "Because you didn't get indoctrinated." And that's allowed me to ask different questions of the research, of the literature that's out there. And it's been a fascinating experience on the heels of 32 years of working in hospitals and hospital consulting firms, to then step out of that role and become a self-taught mineral expert. I don't have the good housekeeping seal of approval of any particular program. I've just relied on the literature and the good fortune of passion and drive and the staying power of over 10,000 articles now. I've learned a thing or two. I wouldn't say that I have total recall of 10,000 articles, but I have a lot of recall. and one of my gifts as a consultant was pattern recognition. I was able to take data and extrapolate it, take information and massage it and synthesize. What is this really telling us? And so I've done that over the course of the last 16 years and have assembled a very unique perspective of how does the body really work. You have two largely competing paradigms. There's attack the guest and strengthen the host. Attack the guest is the more conventional, let's throw a chemistry at it, let's throw a drug at it, try to kill that pathogen or that toxin or whatever it might be. But when you look into the millennia-old healing traditions, most of them are based on what can we do to strengthen the host. And that's really what I think it's from that philosophy that the root cause protocol flows out of. Let's make sure we have the right nutrients in our diet and in our supplement routine to allow the body to express its original intent. Go back to original factory settings and allow the body to express the energy.
Regain homeostasis.
Yeah, to get homeostasis. That's exactly right. And so we ignore the enemies and we ignite the energy. And it's a very different way of thinking about it. And I think it's refreshing, but it's also empowering. Individuals realize I can do this. And that's really the driver from the get-go has been, how do we democratize healing? How do we put the individual back in the driver's seat and draw on practitioners as a resource, but not necessarily as the be-all and end-all? So it's been a fascinating journey over the last 16 years.
Yeah, no, I've listened to podcasts and I've obviously gone through your books, and I can attest just to the way you think through problems, and it's just a delight to see that how you sort of, how do I say it, sort of restructured the paradigm. I mean, we were talking about that a second ago off air, and I think you've just created a whole new narrative or again paradigm to start thinking through these sorts of problems. And yeah, when you were speaking, the two terms just sort of came to top of mind, or one, I suppose, and that is this sort of idea of terrain and germ theory.
Exactly. Right.
I was about to say, do you sort of subscribe to that model? And it's a little off track, and maybe not quite the question I was going to start with, but do you feel that there is an aspect or an element of correctness to that?
Oh, absolutely. I think it's a foundational philosophical divide. And what you've got is, we're going back to the debates between Béchamp and Pasteur. And for those who don't know, um, Pasteur's PhD was a correspondence course in pharmacology. And Béchamp was the most decorated scientist in Europe. He had an MD and 2 PhDs, many, many awards. And of course, Béchamp was taking the position of the terrain theory, and Pasteur was focusing on the particle, the pathogen. And that's where the moneyed interest was on the pathogen. But I think the part that I think it's a nuance of Béchamp about the terrain theory is he wasn't talking about, sometimes it's referred to as the field. He was talking about the energetic field. And I think he didn't say it that way, but as I've studied his work and studied the debates, I think that's where we have this conflict of if you put an electron microscope on a pathogen and you ignore the energetic field that it's finding itself, then you don't understand the problem. And the pathogen is a reflection of what's the state of energy in this tissue. And the fact that the pathogen can morph from bacteria to virus to fungus to parasite, it's like, that's mind-blowing to think about. But it does as the pH changes. And it's just, and what, so the thing is, what does pH stand for? What's potential for hydrogen? Well, actually, it's potential for oxygen. And the really critical issue is when oxygen is available and it can be activated by copper, you make energy, and the peak of energy production is at the pH of 7, because that's when O2 is most available for activation. But if the pH starts to drop or rise, the energy yield drops off. And that's when the pathogens wake up. So I think that whole concept is a critical foundation for understanding where is all this unrest in our body coming from, this lack of homeostasis. It's lack of energy. And we're, to me, it's that simple.
Yeah, and thank you for setting that up. That was the perfect segue into my next question, really, which is to sort of maybe take a deeper dive into why you think we are facing such a high prevalence of chronic disease in society today. Now, I believe your core thesis is that disease stems from oxidative stress due to mitochondrial dysfunction caused by mineral imbalances. I think, anyway, I could be off the mark. Could you explain this a little more? We've got a pretty savvy audience, so the details are good-ish, just as long as we don't start talking about sort of ion channel polarization or anything like that. But up until that point, I think we're golden.
Yeah. No, I think it's the catchphrase is mitochondrial dysfunction. Okay, that's like saying, okay, we're on planet Earth now, and we want to climb a mountain. Well, we got to get a little more specific about where we're headed, and the reason why we're on this planet is because of copper. It's not complicated. Prior to copper's ascension, prior to the great oxygen event, the world was dominated by iron and sulfur. And cyanobacteria began to engage in photosynthesis with this bright, shiny object in the sky, Why that started, I don't think anyone's really been able to identify the origin of that. But when that took place, oxygen began to be given off by those organisms that went into the primordial sea. And then when the seabed filled up, it would pop into the atmosphere. Now we exist right now with about, 21% oxygen in the air. And it's fluctuated over, over the billions of years, but it's now we're hovering around 21%. But when the great oxygen event took place, allegedly 3.4 billion years ago, I'm always amazed at the astrobiologists and their precision for identifying these dates. But when that happened, when there was 0.1% of oxygen in the air, it wiped out 99.9% of life on the planet because it was all anaerobic. Oxygen, it's a very reactive element. It's the second most reactive element after fluorine gas. It loves to play with electrons, especially if those electrons happen to be involving iron. And copper's gift to the planet was the ability to work with iron and oxygen at the same time and not create static, not create a reaction. And and out of that situation was born several critical chemicals. One of them was an enzyme, the classification is called multi-copper oxidases, MCOs, and probably I would argue is probably one of the most important, is called cytochrome C oxidase. And that's obviously what's running complex 4. And that's where oxygen gets turned into 2 molecules of water at the pH of 7, critical pH of 7, because that's the optimal peak of energy production, to allow ADP, 3 ADP, to go over to complex 5 to become 3 magnesium ATP. Now, the part that's a little nuanced, and it's very important for people to understand, is that cytochrome C oxidase is like the stove in your home. What's a stove made out of? Iron. It's made out of steel. And cytochrome C oxidase has a lot of iron in it, a lot of iron sulfur clusters. Actually, there's two, I shouldn't say iron sulfur clusters, there's 2 heme groups. Forgive me, 2 heme groups. But there's also 3 copper atoms. And so think about your stove at home. Does the stove know what's for dinner? No. Does the stove know what temperature to put the oven on? No. Does the stove know which burner to turn on? No. And the world of convention has put a spotlight on the stove of the mitochondria and completely ignored the chef that runs the show. And in this case, it's cytochrome C oxidase, and it has a celebrated function of bringing in 4 hydrogen atoms, or protons, 4 electrons, and voila, in one step, creating 2 molecules of water. That, I think it's the most important chemical reaction on the planet, releases the ADP at that point. And you don't hear people talking about that in the world of convention. They love to talk about mitochondrial dysfunction, and they don't talk about the 55 or more solute transport carriers, many of which are energy dependent. They don't talk about sideroflexin, which is a really important transport mechanism for iron.
I'm not familiar with that. Ceruloplasmin?
Sideroflexin.
Okay.
I just learned it last week, Rob.
All right. It's brand new to me. I've never heard the term before, I must admit.
Well, it's going to make your toes curl when you find out what it does.
Awesome.
And what it does is it lets iron into the mitochondria.
Oh, wow. Yeah, maybe not.
Right? And so then we've got sideroflexin coming in, allowing the iron in, and then we have what are called ABC transporters, ABC B8 in particular, but there's a whole series of ATP transporters that are pushing iron out. You got to get what people don't. I don't think people fully understand is that if the mitochondria starts to accumulate iron, which its tendency is to do, the energy production collapses because the oxygen is not available to be activated to turn into water. And you'll hear about hypoxia. You've heard that term. Well,
Definitely, without oxygen.
there's three different forms of hypoxia. There's altitude. We're at the top of Mount Everest. The air is very thin. It's kind of hard to find oxygen up there. There's a second form called pathogenic hypoxia, and that's neutrophils. Those are the Marines of the immune system that are using oxygen as a bullet, turning it into an oxidant to kill the pathogens. Very important part of our immune system. That function is copper dependent, by the way. That respiratory burst function is copper dependent. And then the third form of hypoxia that no one talks about, I've only seen it in one article, is functional hypoxia. And what it really means is because of a lack of bioavailable copper, and I think it might also involve retinol, which we can get into that as well, the oxygen isn't available to be activated. It's become an oxidant. It's become hydrogen peroxide, or it's become the hydroxyl radical, or it's become superoxide would be the starting point. And
all of these, of course, being free radicals that then cause various types of cellular distress throughout the body.
Absolutely. And we have mechanisms in our body to neutralize them, but they're all copper dependent. And that isn't openly discussed in the literature, except in certain schools of thought.
Yeah, that was going to be my next question, actually. Copper's a role as in support. Well, I know we'll get into this. Maybe we can touch on the ceruloplasmin a bit later, but maybe copper's direct role to support sort of mitochondrial antioxidant activity. I believe, and correct me if I'm wrong, it's involved in the production of mitochondrial SOD or superoxide dismutase. Is that correct?
That's exactly right. So you have the beauty of copper. It's a multifaceted, many-dimensional mineral in terms of what it does, but it's creating energy, right? And it's killing the enemies. One of them being, or it's another way to think of it is creating energy and then clearing exhaust. Those oxidants, those oxygen molecules that have become altered because of electron chemistry, aren't available to be activated and become two molecules of water. And so copper's gift in terms of our life is the capacity to neutralize those oxidants through superoxide dismutase would be one of the most important. And it's clearly involved in mitochondrial production or neutralization of mitochondrial superoxide, but also in glutathione peroxidase and catalase. I mean, the world is just a flurry of thought now about ferroptosis. Everyone's talking about ferroptosis, but there's a very easy way to stop it with ferroxidase enzyme from ceruloplasmin or glutathione peroxidase 4. And not a lot of people make that acknowledged impact.
Yeah. Just for the audience, would you mind breaking down ferroptosis? It can be quite a complex topic, and I know that will probably sort of might lead us to the vitamin A discussion as well. But yeah, would you mind breaking down what that term is and this idea of sort of iron-associated sort of apoptosis, I suppose, or pre-programmed cell death.
Exactly. It's iron-directed apoptosis. It is the generation of lipid peroxides. Lipid peroxidation those are very powerful oxidants that are going to affect the membrane integrity. They're going to affect proteins. It's going to create carbonylation, would be the technical term. And it's also going to lead to the dings of DNA inside the nucleus. The relationship to the iron, again, we're going back to sideroflexin. And if there's too much iron coming in and it starts to react with the membrane, well, you're going to, again, it's a chain reaction. It isn't just, what it's like a pinball machine. And that iron is the steel ball moving all around the machine, and it's got to be stopped. It's got to be neutralized. And one of the most important mechanisms to stop that lipid chain reaction is vitamin E. It's the chain breaking antioxidant that no one seems to talk about. And there's a lot of concern, as you well know, about PUFAs. Stop eating PUFAs. It's like, well, wait a minute, let's back up a step. What are our cell membranes and organelle membranes made out of? Well, yeah, they're made out of PUFAs. And
control the oxidation.
Absolutely. And so what is the match to the fire of lipid peroxidation? It's iron. And that's documented in the literature. I think it's 2008 and 2011. It's just the scientists know this deeper level of truth, but it's not being brought to the attention of students in the classroom about how the body really works.
It really is an Occam's razor, isn't it, at the end of the day? The simplest explanation normally is the correct one. We're sort of wrapped up in PUFAs or seed oils, to be a bit more colloquial about it, that we're not sort of wondering why they are becoming an issue. I mean, historically, they shouldn't be. I mean, it's not like through our evolution, nobody has ever consumed seed oils, and up until recently, they've never really been a cause for concern. And when looking at these sorts of diseases that are metabolic in nature, yet all of a sudden people are happy to sort of point to them as being this root cause issue, when fundamentally they're fairly benign in of themselves. It's the oxidation of them that's a problem. And as you've correctly pointed out, that's being driven by this sort of this mineral imbalance, this copper iron imbalance, that's sort of leading to their sort of excessive oxidation. I suppose while we're on the topic of oxidation, it would be great to sort of at least have an initial sort of chat into the whole world of ceruloplasmin. I think for those in the know, I think people see it as potentially a copper transporter. I might be correct, but it plays just so many more roles in that. It's, as an example, it's an antioxidant. Now, I know this is something we chatted about off air, but it's almost central to your hypothesis in a sense. Could you sort of, or at least briefly, just sort of work through the ceruloplasmin story for the audience?
Can't wait.
There we go.
It's a protein that was first identified in the 1940s, beginning of 1940, 1941, but it's the consistent reference is 1948 by Holmberg and Laurel, and they had broken ground, and they were the ones that had done the most research on it. And it's a protein that has 1,046 amino acids. It's not the biggest, not the biggest protein in the body, but it's one of the biggest.
Yeah.
But what makes it unique is its composition of copper inside that protein. And when it was first discovered, there were 8 atoms of copper. And from 1948 to like 1975, there were 8 atoms of copper. And then mysteriously in the 1970s, the literature started to shift and say, well, actually, it's only 7 coppers. A very mysterious change in the number. And you say, well, they must have had better diagnostic technique. No, that's not it at all. And so then for 30 years, it was 7 atoms of copper. And now in the around 2000, it became 6 atoms of copper. All the recent literature refers to ceruloplasmin having 6 atoms of copper. So put it in the context of your car. If you've ever driven a V8, you know how much power a V8 has. You take 2
Yes.
cylinders out of the V8, it doesn't ride the same. That's a completely different source of power, and it neuters its strength as a car. Well, that's what's happening, I think, to ceruloplasmin. Some of the most revealing research was done by 2 different teams. One was a team out of Harvard, Scheinberg and Sternlieb. And this was in the 1960s. What's very entertaining about that research is that here we have 2 preeminent hematologists from Harvard Medical School doing research at an AT&T Bell Laboratory in upstate New York.
Mm-hmm.
Why were they studying human protein metabolism of ceruloplasmin in a phone laboratory? I just find that utterly fascinating.
Yeah.
But there's a series of 9 studies that they published that revealed a lot about what ceruloplasmin is doing. And then some more critically important research was done by Earl Frieden, who was kind of the dean of metal biology for decades at Florida State University here in the U.S. And he, in the late 60s, 70s, and 80s, did a lot of studies about what is the nature of this protein. And what began to emerge is that, in fact, there's 15 different substrates for ceruloplasmin. It isn't just iron. It worked with copper, it works with oxygen, it works with phenol groups, diphenyl groups, amine groups, biogenic amine. It's staggering what it's capable of doing. All of the catecholamines react with ceruloplasmin. And what it means is that ceruloplasmin has a unique ability to work with those substrates to change their structure and function to make them beneficial. Turn on things, turn off things. And I would argue it's one of the most important mechanisms of intelligence in our body. And one example of the power of this is that some interesting numbers. 36% of the Earth's composition
Mm-hmm.
is iron. That's a lot of iron.
Yeah, it is a lot of iron, yeah.
So over a third of the Earth has some manifestation of iron. And according to the latest research of the World Health Organization, to the extent that we can trust it, 27% of Earthlings, that's a quarter of the people on the planet, are anemic. That's a lot of people. That's 2 million people who can't metabolize the number 1 element on the planet. And in 1971, Dr. Frieden wrote a very important article about ceruloplasmin. And he was doing research to see what happens as the activity level of the ferroxidase enzyme dropped. And so what we have to do is back up and say, so we've got a ceruloplasmin protein that can express itself in 9 different enzyme functions. That's a very unique capability to have. The whole basis of modern pharmacology is 1 gene, 1 protein, 1 function. Well, ceruloplasmin is 1 gene, it is 1 protein, but it's many, many functions. Some say it might be as high as 20 different functions. I'll keep it conservative and just say 9, because I can identify those 9 enzymes. And then we find out that the one that they always talk about in the literature is ferroxidase enzyme. And what it's doing is it's oxidizing ferrous iron plus 2 into ferric iron plus 3. And in that ferric form, it can be either loaded into ferritin for storage, or it can be loaded onto transferrin for transport back to the bone marrow so we can make new red blood cells. Very, very important. Because we're doing that, we've got to replace 2.5 million red blood cells every second of every day. Those are big numbers. I have a friend who says, Morley, there's only three numbers, 1, 2, and a lot. When you start talking about 2.5 million per
Millions,
second,
yeah.
it's just people look like I
Staggering
don't
amounts.
understand that, so it can't be that important. That's usually what people will say. I don't.
Yeah.
I can't process that. But that's one of the most important aspects of our body is that perpetual replacement. And the ceruloplasmin expression is something that Dr. Frieden really studied. And when it went down to 10% of ideal function as the ferroxidase enzyme, there was still healthy hemoglobin production. When it went down to 1% of ideal, it was still functioning, but it wasn't at the right level. And what he asserted was that after 7 days of zero ferroxidase function, that's when anemia began to present in the animals. Anemia is lack of sufficient hemoglobin. That's the real clinical definition of anemia. It's not red blood cells, it's lack of hemoglobin in the red blood cell. And
is that what's then potentially driving the iron deficient anemia?
Exactly. And so it took 7 days of zero ferroxidase expression to then trigger the expression of anemia. And here's the catch. This is very important. Low iron in a blood test does not match up with high iron that's stuck in the tissue. And that is ferroxidase's gift to all life is the ability to recycle iron. That's what it's trying to do as we're replacing 2.5 million red blood cells. We've got to break them down, got to release the iron, got to get it transported back to the bone marrow. And there are a lot of steps in between, but this is completely lost in a world that is preoccupied with dietary iron as opposed to recycled iron. And one of the most important statistics as it relates to this whole dynamic is that only 1 milligram of iron needs to come through our diet. 24 of the 25 milligrams that are needed every day to make that 200 billion red blood cells every 24 hours, 24 of those 25 milligrams comes from the recycling system that is run and regulated by copper. And so you probably have heard the phrase, the simple lie versus the complex truth. Everyone wants the simple lie. It was Alex de Tocqueville who said it in France, but centuries before him, it was Lao Tzu in China. People would always prefer the simple lie than the complex truth. It's just easier to accept. Well, the simple lie is that people are anemic. The complex truth is I have access to articles that, to my satisfaction, prove that there's 9 copper-dependent enzyme functions that contribute to, quote, anemia that have nothing to do with iron. And it's absolutely fascinating that the world has been captivated by this idea of low iron and has very little understanding or awareness that copper is running the show. Copper is the general and iron is the foot soldier. And that's not how it's presented in the literature at all.
Yeah, definitely the other way around. And for the most part, people are more than happy to just try and treat these conditions, ironically enough, by, supplying the body with more iron. I suppose that's the great irony there, isn't it?
Absolutely. I mean, and it's just the world does not understand that copper and iron are interdependent. And if I were king for a day, if I had a magic wand, I would require all studies of iron to involve copper metabolism and all studies of copper metabolism to indicate its impact on iron. We've got to get beyond and get people to realize this interdependence that these metals have in bringing homeostasis that you were referring to at the very beginning.
Yeah, there's sort of, again, it's a common theme in sort of biology as a whole. Everybody sort of gets stuck in a sort of a siloed, in their own silo, don't they? Irrespective of where you are, and you sort of end up missing, what's the expression? You end up missing the woods for the tree, something to that extent.
lose sight of the forest for the trees.
There
And
we go.
we had the pleasure of spending an evening with Douglas Kell, who's a
We did.
world-renowned iron expert. And when he was at University of Manchester, his best friend was Garth Cooper, who was in the office right next to him, who's a world-renowned copper expert. And according to Dr. Kell, they never talk shop. I'm like,
Wow.
what? But I'll take him at his word. They just enjoyed each other's company. They'd go to the pub and let off some steam. But it's like they didn't talk about how their two research worlds were interwoven in a very intimate way. And that's the challenge we've got around the globe, is getting more people to be aware of your curiosity and this. So what are we missing? What's the part that we don't seem to understand? And I think it's this idea that somehow, in my way of thinking, copper's running the show, but we're taught that it's toxic. And when you get into the real bowels of human metabolism, especially, it's undeniably central to our health and physiology. It makes no sense any other way. And it's just not taught that way in any school of nutrition or any kind of doctor's school, whether you're allopathic or homeopathic or osteopathic or naturopathic. Copper's not part of their vocabulary. And as I was noting before we started the formal part of the conversation, I've seen 5 textbooks, not one mention of this protein that you're curious about, this ceruloplasmin. And what's also important for people to know is that it's subject to modification. It can get what's called denatured, so that in the presence of ascorbic acid,
Or citric acid.
it changes, or citric acid, it changes its structure and the copper gets released. In the presence of high glucose, blood glucose, when blood glucose gets above 120, it denatures ceruloplasmin, the coppers come out. And so everyone is taught to fear this unbound copper, but copper's concentration in the blood is 1% of the copper's in the blood. 99% is in the tissue. And if the copper is rising in the blood, it means it's missing in the tissue. And it's the absolute inverse of iron, that when iron is low in the blood, it means it's stuck in the tissue. And it's just getting people to see the symmetry of that and how that gets represented in the testing. But we don't have access to perfect testing, because at least here in the States, we're not allowed to test for the ferroxidase enzyme function. There are only a handful of countries in the world that will even allow it.
I don't think I don't think I've ever even seen it on a lab request sheet, to be honest. I suppose, speaking of testing, you're a proponent of a hair mineral analysis in that respect. Could you sort of briefly just walk us through that? And then maybe we could sort of transition into your protocol specifically.
Sure. Been working with the hair tissue mineral analysis from the beginning. And my mentor in those early years, was a gentleman by the name of Rick Malter. He's a clinical psychologist, and he had been using HTMA data for decades, maybe three and a half decades. And he was very quick to point out to me that there's really 2 questions that can be answered in a hair test. One, is this person under stress? Very important, because it will reveal itself very quickly in the hair test. And secondly, can this person mobilize energy in the face of their stress? And he developed something called the Malter mix to normalize the results, so that you're seeing what's the actual magnesium relative, and what he was doing was relative to its ideal, actual calcium relative to its ideal, and so forth. And so the Malter mix is a great way to level the data from the hair test, because it's very easy to be overwhelmed by that trees versus forest dynamic. And I think when my thinking really began to change about the hair test was when I realized that you can't measure ceruloplasmin activity or level in a hair test. We can measure the mineral copper, but we can't get to its protein that really runs the show. And so that's when I began to move into the world of blood testing. And it's not perfect because there's so many restrictions on what we're allowed to test in the blood. But it presented a completely different perspective of copper as it relates to its influence in the body. And so the hair test, I think, it's an absolutely vital source of broad information, because what you're looking for is to what extent is this person's mineral expression all over the map. And the more it's all over the map, the more stress they're dealing with. And then it's very easy to reveal, are they making energy through the adrenal ratio and the thyroid ratio, which is kind of fun.
That's interesting.
Yeah, to see that. And to me, that's where the rubber hits the road. Is it really relates to a famous quote by, at least I think it's famous, quote by Mark Hyman about stress. Stress is the body's inability to make energy for the mind to respond to its environment. I think it's one of the most profound definitions of what stress really is. And what we've got to be able to do is, when you have stress in your world, I guarantee you're going to have oxidative stress in your body.
Definitely.
So then we're into functional hypoxia. Suddenly the oxygen is not available to be turned into water. We can't make energy. And then that's when all the symptoms begin to surface, whether it's toxic symptoms, pathogenic symptoms, heavy metal symptoms. That's when everything begins to flourish, is when we can't make the requisite amount of energy. And so when you look at a hair test and you see this erratic picture, which is very often the case, what you really know is that person's under stress. Can they make energy? Most can't. How can we solve that? That's what the root cause protocol is all about, is how do we reignite the ability to make the energy so we can respond to the stress and have resilience in the face of that stress.
Perfect. Morley, I could talk about biochemistry with you all day. However, I have a feeling the audience would like something maybe a little more practical. So I'd love it if we could sort of maybe dig more into what you're probably most well known for, which is really your root cause protocol, which you've developed on sort of the back end of the research that you've done for the last, nearly 2 decades at this point. Could you walk us through the protocol? I've got a bunch of questions of my own, but which I'll interject, and I'll interject along the way when they come up. But in a nutshell, how is your approach to helping people overcome various health challenges, working?
Having come out of the conventional medical model, where there's disease everywhere, and I was involved in doing forecasts of where were the disease indexes going to go, and they were all hockey sticks waiting to take off. I realized that there was something fundamentally wrong or something misunderstood, and so that's what really pulled me into this whole quest of, so how are we really designed to work as a species? And I started out focusing on magnesium because it's the first mineral lost to stress. And I was influenced by Carolyn Dean and Mildred Seelig and Jean Durlock and a number of other famous researchers who were able to connect the dots that magnesium is the first to go, quickly followed by the B vitamins when we're under stress, because they're water soluble and they disappear very quickly. And so that morphed into trying to understand, what are the biggest sources of stress? And then I find out that there was an article by an Italian team of researchers that revealed that iron stress is the greatest stress on planet Earth. I went, wait a minute. And then I began to sync it up with the research about the great oxygen event. So I got iron and oxygen. And then I really began to delve into, so how do we stop that? And it's copper. back to our earlier discussion. Copper, when it's bioavailable, when it's usable, when it's attached to its appropriate spectrum of enzymes, is able to neutralize the static and create the energy. And so I set out to, so how do we make copper bioavailable? We got to have ceruloplasmin. And I was reading an article by Ray Peat, who's a recognized.
Since passed on, unfortunately, yeah.
Yeah, right. But this goes back 10 years ago. And it was a wonderful article about iron overload. He wrote several articles, but this one particularly was focusing on iron overload. And at the end, towards the end, he said, to my knowledge, no one has ever developed a recipe to increase the production of ceruloplasmin. That was a red flag in front of this bull. I said, well, that's what I'm going to do. I'm going to create that recipe. And at the beginning, it was, I knew that it was going to be important to have things to stop doing and start doing, in large part because of a conversation I had with my oldest son, who's an engineer. He said, Dad, you gotta tell people what to stop doing. You got to tell them what to start doing. I went, oh, okay, stops and starts. And so at the beginning, there were 2 stops and 2 starts. And the 2 key principal stops were stop taking iron supplements and stop taking vitamin D. You can imagine the heretical stance I was taking, but we've weathered that. Go ahead.
Sorry, I was just going to say, what for the audience is the issue there with vitamin D specifically?
Well, it's just what we're proposing is a balanced blend through cod liver oil to get both retinol and vitamin D. Don't take a vitamin D supplement standing alone.
In isolation.
Yeah, it's just not advisable. And there's a whole masterclass that I have on the downside of vitamin D that people are welcome to purchase online. It's not very expensive, but it goes into about an hour and a half of here's what you don't know about vitamin D. There's a whole other side to it. And so it's evolved over the, I guess, probably 10, 12 years. It's evolved into about a dozen stops and a dozen starts. And for those who are interested, just go to the website rcp123.org, go to the resources tab, and then there's a starter guide. But it's the latest version of our getting people started on the root cause protocol that is printable. You can put it up on your fridge or wherever you want to post it so you can keep track of it. But all you have to do is donate your email address, and we'll send you the record of the guide itself. And it's really been met with a lot of gratitude. A lot of people are very grateful for it. And it lays out not just the stops and starts, but there's a schedule. What should you do when? And it answers some of the basic questions. There are other resources. You can get the RCP handbook, which is 84 pages long. So the starter guide's 12 pages. There's an 84 page. These are both free. There's no charge. We're giving away the answer, and it just takes discipline to work with it. And it takes a willingness to step out of the box of convention that tells you, do this, do that, when in fact, no, you don't want to take ascorbic acid. You don't want to take calcium. You don't want to take vitamin D alone. Okay to take cod liver oil. And then the flip side being, it's important to take whole food vitamin C. It's important to take magnesium and make sure you've got minerals in your water and so on and so forth. And what we found, it's absolutely fascinating. People who just do the stops, when they do those dozen or so stops, they find that they actually start to feel better. And then as they start to adopt, we have a phased introduction of the starts. That's when That's when the magic starts to happen. And it's across the board. It's the whole concept of the root cause protocol is profiled in my book, Cure Your Fatigue. And again, the basis of that is there's 20,000 symptoms that are profiled in the Merck manual. That's the Bible of medicine, is the Merck manual and the description of all those 20,000 problems. But what do they all begin with? Oxidative stress. The oxygen is not being turned into water, and when that happens, it sets the stage for symptoms. And depending upon where it's happening, whether it's in your mind or your brain, I should say, or your kidney or your elbow or wherever it happens to be, it's called something different in the Merck manual, but it's all one origin. And it's the three-ring circus of the world, as I know it, is copper and iron and oxygen not playing well together. And when they don't, they create this spectrum disorder that we call chronic disease. And so the RCP is really designed to empower people to take control of their situation. And we have recommendations about an ancestral diet. We don't get into the fevered pitch of ketosis versus paleo versus whatever. Follow the tenets of Weston A. Price. He was a very brilliant scientist, gifted dentist, but he was a very, very profound scientist who studied what did the original communities eat to have perfect teeth. And why was he obsessed with perfect teeth? Because he knew that when we're a little fetus, we start out with 32 buds, and the 32 buds split and become 64 buds, and 32 become our teeth, and the other 32 become our spine. And he knew that perfect teeth meant perfect spine meant perfect health. And so I just think it's important for people to realize that there is this legacy of recommendations out there. And we can get into all sorts of religious wars about nuances of the food system.
The minutiae, yeah.
Yeah, it's exhausting, as you well know. But I think that the goal is to try to simplify this message and simplify the process so that people can, in fact, regain mastery of their physiology as we are designed to do. I believe that we are sovereign individuals, and we're meant to have this independence of action, independence of thought. And when you eat the right foods and you have the right supplements, it enlivens the innate healer within us. And I'm not suggesting that we no longer have stress. We will always have stress as long as we're above ground.
Yeah.
But what we can do is neutralize that stress with a level of proficiency and efficiency when the minerals are optimized and we've got an ability to curb the stress that's around us, whether it would be environmental stress, physical stress, biological. I mean, there's many different forms, but the body has this capacity to heal itself under all those types of conditions.
Yeah, no, it's very adapted sort of regulating itself and refinding homeostasis when the opportunity presents itself. But as you've alluded to multiple times, the moment there are breaks in mitochondrial efficiency or high levels of oxidative stress in the body, then it's essentially almost, well, very literally putting out fires, and it's not in a position to really regain its fundamental structure. So essentially, what you're saying is that it's just it's a tad more complicated than just popping a couple of capsules of copper glycinate every day, but it's still very doable for the average person. Just a sort of a quick tangent. Speaking of healing modalities, what are your thoughts on molecular hydrogen? It's doing the rounds at the moment, and it does seem to have quite a lot. It does seem to be quite effective, both from supporting mitochondrial function directly and through its sort of actions as a sort of selective antioxidant. Do you feel that that potentially has a place in, well, either in your protocol or in health in general?
No, it's a wonderful question. And I'm often asked about molecular hydrogen or ozone therapy or other add-ons, if you will. I tend to take a very conservative stance. Um, Most of those modalities are designed by scientists or practitioners who have no deep knowledge of copper metabolism and what the um, enduring role of copper is on the planet. As it relates to the hydrogen, what's the primary source of hydrogen protons in our body? It's the mitochondria. Complexes 1, 3, and 4 are pumping hydrogen constantly, right? That's their job. Now, what most people don't know, I think the literature is very selective about what it reveals, but complex 1, 3, 4, and 5 are all copper dependent. I mean, 99% of articles will say that it's only complex 4, but there's compelling research around complexes 1, 3, and 5 that no one likes to talk about because it begins to spoil the show.
The
So,
inconvenient truth.
yeah, exactly. And so I'm well aware of in a lab setting that hydrogen can neutralize the hydroxyl radical. I think it's a little bit of a stretch to say it's going to happen in a biological system because we happen to put hydrogenated water into our body. I, I just, my limitation is I'm not a chemist, I'm not a biochemist, I don't pretend to be a biochemist. I'm trying to think like our ancestors did. How do I maintain a level of vitality through my diet, through my supplement routine, manage my stress, both what I can control in the outside world and my inside world, and just go about my business. I get nervous when people start to reach out for different modalities because it sets the stage for two things. One, I don't think we fully know what those hydrogen protons are doing. And the other side of it is when you start to adopt all these different approaches, basically what you're telling the body is, I don't really trust you. I don't think you really know what you're doing. Let me give you an inducement. Let me enhance what you're doing. And it sounds silly, but I think the body says, well, why are you asking me to get involved if you think you know the answer? And the problem we've got is that you and I are engaged in what I would call a very switched on conversation, exchanging information at about 2,000 bits per second, that's pretty fast. The part of the brain that runs our body, the hypothalamus, thinks a million times faster than you and I can talk. And I think it's a very bold thought to say, well, I read this article on the internet and I need to do this. I think the body knows what to do. It lacks the nutrients to make the energy to run the immune system to regulate homeostasis. And I just take a very conservative stance that I'd rather let the body run itself than me start. And again, there are recommendations in the RCP, but these are the stops are to eliminate what never existed in the food system before, and the starts are to try to restore normalcy and sanity in the food system that our ancestors had full advantage of. That's very basic, and I think it's also we could easily add 50 other items to the RCP. Well, then we have a compliance issue. Just getting people to do 12 things.
The basics.
Yeah, just doing the basics. And so there's a trade-off. And so I recognize the popularity of the hydrogen water, but I had the good fortune of spending a day with a world-renowned consultant to the natural food industry. And During the course of that day, um, his first name was Michael. He said, Morley, people always ask me, Michael, what's new? What's new? And he said, I've learned to ask what's enduring. And he said,
Yeah, it's a good way to look at it.
He said, I would strongly advise you start focusing on what's enduring. And it was really out of that conversation that I stumbled into the research about the great oxygen event. And that completely changed my understanding of the problem and the realization of what copper's role is on the planet. And I would argue that people relying on hydrogen water are probably copper deficient without the benefit of testing, because the copper knows how to pump hydrogen. The mechanism of making stomach acid, the mechanism of recycling red blood cells requires acidification. Well, that whole process is copper dependent. And what's it doing? It's pumping hydrogen into these vacuoles to allow change to take place. These hydrogen atoms don't come from Mars. They're coming from copper enzymes that are designed through the ages to support our physiology.
That's a very concise answer, not one I expected, so thank you. Morley, I've got some follow-ups to you, well, maybe some other viewpoints I'd love to get your opinion on. But before we get to that, or those particular viewpoints, I suppose, I'd love to kind of get your thoughts on trauma and the central nervous system and dysfunction there in general. It's something I'm starting to see more and more when I either work with people or I suppose myself as well. But the fact that people, for the most part, when they're following a protocol, often make at least some level of improvement. But unless they actually start to deal with a lot of the emotional side of things, the past traumas they've been through, they never really start to heal. Could you weigh in here? And do you have any thoughts or on any techniques that you feel are effective at helping people to overcome trauma? Assuming you think it's an issue to begin with.
Oh, it's very real. And again, I go back to my mentor, Rick Malter. He wanted to make sure that I understood that the hair test was a window into the person's stress profile. And very, very important. We're all under stress. You and I are both under stress. We're having a lovely conversation, but everyone we know is under stress. Everyone we don't know is under stress, because that's the human condition. And when I started this work many years ago, I was very Newtonian. I actually have my grandmother's brother's name was Newton Matthews. Our ancestry is back in England. And family legend has it that we're related to Sir Isaac Newton, although he didn't have any children. So it's one of his collateral, brother or sister or something. Somewhere we're related to Sir Isaac Newton. But I was very Newtonian in my view. There's some physical event missing. It's a nutrient, it's a vitamin, something is not there. Over the course of 16 years, I've become very quantum, realizing that at the very heart of all of this physical imbalance is emotional unrest, which is very energetically driven, as you well know. And I think it's very, very important to address that. And so for years, we've been talking about the importance of dumping fear, because that's the granddaddy emotion of all, is that when you have a chronic condition that doesn't resolve, doesn't respond to the recommended path, you begin to doubt yourself. And you begin to think, well, there must be something wrong with my body, or I'm not doing it right, or I'm being punished by God, or something to those effects. And so that conjures up fear, and we spell it differently in the RCP, Fe-AR, and that way you see the symbol for iron. And when you're under stress, you become a magnet for iron. Now, we've been saying this for years, but it's only been in the last month that I have definitive physiological proof that it happens. And there's an enzyme in our body that you may have heard of called furin, Furin, and it's what's called a protein convertase. It's a very ancient mechanism. It's not as old as ceruloplasmin, though. It's not as old as the PAM enzyme, which is a very powerful enzyme in our body for making change. But furin has a unique ability to activate the iron hormone called hepcidin. Now, hepcidin is what I call, well, it's not me, it's known as a negative regulator in iron metabolism. And ferroxidase and the ceruloplasmin protein, would be known as a positive regulator. Well, looking back on our childhood, our parents were the positive regulators in our upbringing. A negative regulator would be a SWAT team. And what the world of convention wants us to believe is that SWAT teams are running iron metabolism and not our parents, which makes no sense at all. But the important thing to understand is that when hepcidin starts to flex its muscles in response to furin enzyme that is triggered by stress, and the form of stress that triggers it the most is social defeat stress, otherwise known as PTSD. Go back 5 years ago. The world was engaged in PTSD.
Just a bit.
Just a bit. And so furin was off the chart. Hepcidin, off the chart. Iron regulation was completely gone. And so I renamed COVID. COV stands for copper's vanished. ID stands for iron's dysregulated. And what your listeners need to make sure they understand is that when copper's down, iron takes off inside the body. And so the mind and our psyche will convince us that we're under stress, which is only going to feed the problem of iron regulation. And so that's why dealing with the emotional side of the equation is so important, which most people want to ignore. They want to stay away from their psychological or emotional trauma. And I get that. We've all had some very severe trauma that we want to pretend didn't happen. But when you are able to engage in emotional release techniques like emotion code or body code, integrative processing technique or EFT, it's tremendously liberating because it releases the fear that you've done something wrong. And it then says, as long as you're in a state of fear, you're in a sympathetic state. Well, guess what doesn't happen in the sympathetic nervous system? Healing. You can't heal and run from the bear at the same time. So you have to be in a parasympathetic state. And so that's the dilemma is we're living in a society where most people 24/7 are in sympathetic overdrive. And lurking in the background is this enzyme that no one talks about, furin. It's a very sophisticated audience that would know what that is. And then influencing hepcidin that very few people know about. But here's the most important part, is when hepcidin is elevated and activated, ferritin is low because they ride on a seesaw together. And so who taught me that? Douglas Kell.
I was about to say, because ferritin's an acute phase reactant, isn't it? So that would then make sense as to why it's an acute phase reactant, obviously.
Exactly. Beautifully said. And so what's the one consistent mistake being made worldwide about ferritin? That low ferritin means you're not storing enough iron. You need more iron or an infusion. Well, I don't call them infusions anymore. They're invasions. And it's a very serious problem on the planet that because people don't know that there's 9 copper-dependent enzymes to regulate the production and the recycling of iron in the blood, and all we do is default to, you need more iron. No consideration given to these 9 different expressions of miles per gallon, because you can put if you're having trouble with your mileage, you can fill the car up with gas, and it's not going to get better mileage, is it? You've got to change tire pressure, timing. You've got to change a lot of factors in the engine and in the car's performance to get better mileage. But throwing more gas in the car is not going to solve it. And that's the mentality of most practitioners, whether they're doctors or nutritionists or whoever. They have been taught singularly, you need more iron. It's like, really? That is, to me, that's the foundation of where the breakdown is in healing today, is believing that dynamic that only iron will solve a low representation of iron in a blood test. When in fact, I did this just the other day. I was with a buddy of mine who's very good at AI, and I said, Steve, what are the top 10 causes of anemia that have nothing to do with iron? And what came back was B12 deficiency, B9 deficiency, B6 deficiency, hemolytic anemia, blah, 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 blah. And it goes through the whole beta thalassemia. And then but there were two things that were missing. I said, okay, what are the top 12 causes of anemia that have nothing to do with iron? Number 11, copper deficiency. Number 12, retinol deficiency. And to me, that was very representative of the thought process of convention. I'll give you 10 ones that you know about, but only under duress will I give you the two most important.
Yeah, no, I'm surprised to get with those. I was expecting another answer. I was expecting you to carry on to at least 20, to be honest.
So I was delighted that the AI gods let the cat out of the bag. But the important thing is this concept of stress, of emotional stress, is central to our health and well-being. And everyone's under stress. And again, to reinforce a point that I made earlier, if you have stress in your world, and we all do, that means you have oxidative stress in your body. And if you have this perception that the stress is unresolved or unresolvable, then it becomes PTSD. And then you've just put gasoline on furin, then you've just totally changed the hepcidin ferritin dynamic. And then the tragedy is the people who have the low ferritin get iron supplements, get some kind of enhanced level of iron. And what does that do? It only increases the physiological stress in the body. Because when iron is too high, it has a wet blanket effect on copper metabolism. So then we've totally changed the physiology of the body, which is only going to intensify the perception of stress in the body. I think to me, it's the hidden factor for why so many people are out of balance and have chronic fatigue and just don't have the vitality that they want, is this confusion in the healing circles about what's really behind low iron in the blood, and not any consideration to it's stuck in the tissue, and not realizing that copper is the shuttlecock, if you will, between the two domains.
Definitely. So take a chill pill and let it be called copper. Okay.
Absolutely.
Morley, I'd like to maybe introduce thank you for that, by the way. I'd like to introduce a slightly different perspective if you're open to it. Now I'm a big fan of Joel Greene. I don't know if you are familiar with his work and who he is. He has a very gut-centric approach to health, and health in general. And he also has a fairly, I suppose, multifaceted approach to how one might deal with iron overload, which I'd love to get your take on. Of course, I'm not trying to pit anyone against each other.
Nice.
It's just really I'm just interested in hearing people's perspectives, I suppose, on different protocols and the ways of approaching something. Anyway, he first posits that he first posits that increasing various gut bacteria, like bifidobacteria, for example, with various iron-binding pseudophores, which I suppose for the audience can maybe best be described as well. I suppose literally bacteria produce magnets, can help trap iron and I suppose sequester it at the level of the gut and therefore reduce systemic overload. Do you think there's any value to that statement at all?
Is there any way?
Value to that statement?
Oh,
Would you agree with that?
absolutely. Iron metabolism is a two-step process. We've got to get iron into the enterocyte, and then it comes in in a +2 format. But then it's got to go into a +3 format to either be loaded into ferritin or be exported onto transferrin. And the second step of absorption is transferrin accepting the iron so it can go into the bloodstream and get back to the bone marrow to become new red blood cells. And so when that doesn't happen efficiently and effectively, there will be a buildup of iron in the enterocyte, the enterocytes. And so I think that all of the gut-related distress that's out there, whether we're talking about colitis or Crohn's or just IBS or whatever the mechanism might be, I think it's all iron related due to a lack of copper. And in fact, in the world of veterinary medicine, there's a condition called Jonas disease, Johnses, Jonas disease, and it's identical to Crohn's disease. The conditions are absolutely identical. Do you know how they cure Jonas disease in the animal world?
Copper.
Yeah, but in the human world, the doctors wring their hands, saying we don't understand this Crohn's thing. And it's just, to me, it's unfortunate that people don't understand how iron is absorbed. 2 steps, how copper is essential for the mechanism to maintain that balance. And so the other side of it, though, in terms of gut dysbiosis, is I've worked with hundreds of people who have gut issues, as you can imagine. What do they all have in common? There's an emotional issue they can't stomach.
Yeah, and that's sort of going to affect that migrating motor complex for the most part. I assume there's a copper link there as well.
Well, again, we're back to when you're in a sympathetic state, the 2 hormones that are going to really rise are adrenaline and cortisol.
Yeah, fair enough.
Adrenaline has known properties to increase hepcidin. So there must be there's an adrenaline-furin connection, right? So hepcidin's going up. We know what that's going to do. And what does cortisol do? Cortisol is a very important hormone. It serves many different functions. But one of its least known functions is the ability to increase the production of metallothionein. So in a state of stress, when cortisol gets released, there can be a 4 to 5-fold increase in metallothionein production. And why is that a problem? Because metallothionein binds up copper 1,000 times stronger than it binds up zinc.
Oh, okay.
So then we've just changed the energy and antioxidant dynamics of the body. And so if someone has chronic stress, essentially what's happening is the organism, our organism, is saying, you can't seem to handle the stress. I'm going to power you down.
Yeah. Hence the, I suppose that's sort of something I go on about in every podcast, just about, so I'm sure the audience is ready to kill me at this point. But that sounds like very much like Dr. Robert Navio's cell danger response. I don't know if you're
Yes,
familiar with
absolutely. Yeah. And the other part that I think is important for people to realize is there's a lot of belief. And what's the 3-letter word in belief? Lie. There's a lot of belief that we need to manage our zinc-copper ratio. No, you don't. You need to get zinc in your diet. It's a very important mineral. But I think we are facing a severe shortage of copper in the food system because it's not in the soil, because of modern agricultural practices. But what people need to understand is that zinc is a perfect activator for metallothionein. So when you're gobbling down 45, 50 milligrams of zinc, you've effectively taken copper offline. And the part that people may not know is that zinc has a known ability to block copper uptake at CTR1, copper transporter 1. It has a known ability to kill the cytochrome C oxidase function. And it has a known ability by Dr. Deuce down in Australia in 2010 to kill the ferroxidase enzyme function, especially in the brain. And so there's a lot of who struck John about, oh, you got to be careful of your zinc levels. And no. The person who raised that issue was a gentleman by the name of Carl Pfeiffer in the 1960s. On the heels of world-renowned scientists like Otto Warburg, Hans Krebs, LVM, Conrad LVM, some very noted scientists studying copper and iron for 30 years. They didn't study zinc. Zinc wasn't even on the radar screen. And in the same way that I would argue that Louis Pasteur was guided in his focus, I think that Carl Pfeiffer was guided in his focus. And he wrote the first article in the 1960s about the zinc-copper ratio. Well, what's important to know is the history of these scientists. And it's in Wikipedia. You can look it up. But he was a principal in the MK Ultra Mine program of the CIA here in the States. Well, when I learn about a fact like that, that invalidates their research. And so I question the veracity of this zinc-copper ratio that runs a lot of thinking in functional medical circles. They get very preoccupied with it. And I don't think they understand the energetics of copper or the de-energetics of zinc. There's nothing about zinc that creates energy. It doesn't make you want to have more vitality. It doesn't. To my way of thinking, zinc is a structural mineral. Copper is a catalytic mineral, and we know we need both. But when you've got to make change, when you've got to make energy, when you've got to neutralize the exhaust, you better have bioavailable copper at your beck and call.
Do you think the copper RDA is even close to being sufficient?
Excuse me?
Do you think the copper RDA is even close to being sufficient?
So in the 1930s, the average person, this is here in the States, but actually there were studies done in China and in UK. Back in the 30s, the average diet would deliver 4 to 6 milligrams of copper a day.
And now we're stuck on 1 to 2 at best.
And then by the 1960s, it had dropped to 2 to 5 milligrams per day. And now worldwide, they seem to be hovering around 0.9 milligram being acceptable. And then Leslie Clavet in 2011 did a study of the average American's diet and how much copper were they actually getting. And if I'm remembering it correctly, 80% of people in the States were not getting 0.9 milligram of copper a day. And he just has a very simple solution. Get more copper in your diet and in your supplement routine. And that's a very unpopular, almost heretical stance because copper's toxic, don't you know?
Yeah, you're going to end up with Wilson's disease by tomorrow morning if you overdo it by 0.5 milligram.
Exactly, right. No, I think there's a lot of hysteria around copper for all the wrong, well, I will say for all the wrong reasons. But the delicacy of copper is the research is out there, but you really have to work for it. And it takes time and discipline, and it takes a willingness to stand up to convention, to question, could there be more to the story? And that's really that's the position we take within the RCP is, yes, there is more to the story, and let's talk about it.
Yeah. Do you have a preferred sort of supplement form of copper? I mean, I know there is one, I believe it's by a company called Global Healing, and they produce a copper, it's a cuprous nicotinic acid, I believe. I have a feeling that you're a fan of copper supplements in the sort of alongside other whole food forms of various vitamins and minerals, such as A. Um, what are your thoughts on sort of standalone copper salts versus copper in, yeah.
When I started this work, I was really focused on more on minerals in general and magnesium. What really woke me up was COVID, that we have a problem, that I think there's a definite shortage of copper in the food system. Um, there's a lot of opinions about it, as you can well imagine. But I was inspired to help a supplement company based here in the U.S. It's called Formula IQ, to make a copper supplement called Recuperate. That's my unending wit. Recuperate. And it delivers 2 milligrams of copper bisglycinate along with spirulina and desiccated beef liver and some boron and turmeric. And it actually comes with or without the boron. I fought the need to do that mightily, and I was willing to run the risk of being called a supplement whore because I somehow named it. I had no influence over its design per se, although the formulator was very quick to get at least my opinion, because I'm not a chemist. But I wanted a solution to the copper problem that I think was definitely aggravated by the time period 2020 through 2023. There was a decided attack, in my opinion, on copper status, other minerals in general, but I think copper in particular. And so, at the risk of sounding like Al Gore, who invented the internet, right?
Yeah.
I think I invented the demand for copper. And
I think you did.
I think I made it fashionable to at least talk about it. Right now, there's probably 50 different products out there, which I think is great. I think, let's raise the tide because it's going to help all boats. I think what's really needed, Rob, is a definitive study to say, is there a pecking order to all these different approaches? I've seen articles that are very critical of the salts, the copper salts. I've seen articles that talk about the value of copper infusions. I mean, the dilemma is what's really missing. If I had another magic wand, I would institutionalize the production of ceruloplasmin and allow that to be something that can be infused in people.
Recombinant ceruloplasmin.
Yeah, exactly.
Why not?
Because it exists. It costs about $200 a vial, a 1 ounce vial, but you have to be a research scientist to get access to it. And the thing is, back in the 1950s, they were curing schizophrenia with 1 shot of ceruloplasmin.
Oh, I did not. I was not even close to aware of that. That's fascinating. I'll look that up.
And so that's work that was done at Tulane University in 1959. Dr. Jensen and 2 colleagues from Harvard Medical School were involved in that. And they were studying 34 patients with schizophrenia. And there was a marked improvement in 30 of the 34 with the shot of ceruloplasmin. Well, what does that really tell us? Well, the origin of schizophrenia is the rusting of adrenaline. Because anyone who has schizophrenia is in a state of fear, heightened fear. And what's the byproduct of fear and adrenaline? It's called adrenochrome. It's a very powerful chemical that alters the thinking in the individual's brain. And why do I know so much about it? Because my dad had schizophrenia. And what was the treatment of choice in Baltimore, Maryland in 1958 when he was when he actually ran away from home, never to come back? Was they were using electric shock therapy, not ceruloplasmin. And the reason why he left home was, if you've ever seen the movie Cuckoo's Nest, you know how unbecoming electric shock therapy is. And he didn't want any part of that because he'd had it done once. So it's just, that's the wound that I carry. And I have the pleasure of knowing how to solve it, but I don't have the pleasure of administering it because it's under lockdown. It's under clinical lockdown, and it's only available for research scientists under very strict conditions. But I can point you in the direction of a dozen studies to prove that ceruloplasmin would solve all the problems.
Yeah. Well, I mean, hopefully through sort of talking about it through on podcasts like this, we can at least raise awareness and create the
Absolutely.
education around it.
Yeah.
I found the study, by the way, just while you were talking, and I'll make sure to link to it in the show notes so that people at least do have access to it. Okay.
It's a very disruptive article.
Just a bit. I will read it in depth after our podcast.
Yeah.
Morley, the last thing I suppose I wanted to talk to you about in earnest today, and this is again something Joel Greene brought up, was this concept of lactoferrin, specifically human lactoferrin. Now, I suppose Joel talks about this as being a master regulator of iron balance and a two-way buffer system, maybe rather than just a one removal tool. And the way I see it, and you're welcome, and correct me if I'm wrong, which I probably am, but it binds to iron reportedly anyway up to 300 times more than transferrin, allowing it to obviously then sequester more iron. It then reduces free sort of redox active, I suppose, iron to limit oxidative stress, and I suppose as a result of that, microbial growth by way of limiting substrate for infections like Candida, which we didn't touch on today, but that's fascinating in of itself. It also seems to release bound iron in the body when it needs to, and it's then able to bind up a surplus. And then finally, it seems to sort of support immune system function, and by modulating, and let me see if I can get this right, macrophage function, which I assume could help regulate iron recycling by way of ferroptosis. Is that correct?
Yes, that's right. Yeah.
So I think I'm piecing this together, at least in some way, shape, or form. But do you think lactoferrin is an interesting molecule in this regard? And I know we're going back slightly to the argument, well, not the argument, the discussion around molecular hydrogen and adding sort of more pieces to this particular puzzle. What do you think about lactoferrin in general, and maybe with these new human-specific lactoferrins that are coming onto the market that potentially have less of an immunological reaction than, say, something that's bovine in nature? Do you think that has a part to play in helping to regulate iron?
No, I think it's a primal molecule to do that. What's one of the highest expressions of lactoferrin on the planet? In unprocessed cow's milk.
Raw dairy, yeah.
Raw dairy. And we're back to Béchamp and Pasteur.
Indeed.
When you pasteurize milk, what happens to the enzymes in that milk?
They disappear.
There's 50 of them.
Yeah.
There's 50 of them that disappear. The two most important, ceruloplasmin and lactoferrin.
Oh, wow. I did not. Okay. I've learned something else new today.
If I know that, do you think they know that? Of course they know that.
Yeah.
And we don't just pasteurize milk now. We ultra pasteurize it.
Yeah. I still find it quite funny that people Look, I think raw milk definitely has its place, but what I find funny is that people will buy raw milk and then put it straight into their coffee. And yeah, your raw milk isn't so raw anymore, is it? It's just milk.
But that's a very, very insightful observation. But the thing is, um, how much how much iron is in breast milk?
None, I assume.
None. That's exactly
Yeah.
right. Do you think there's a reason for that? Well, there is, because the baby, the infant human, doesn't have an immune system until they're 2 years old. What is their immune system? Oh yeah, it's copper driven. It's the liver, but it's copper driven. And during the height of insanity of COVID, I found 52 articles that documented that copper was in charge of the immune system. COV, copper's vanished. ID, iron's dysregulated. And so um, the infant, and what's the level of vitamin D in mother's milk?
Actually, I don't know offhand. Um,
0.
okay. Yeah,
Mother's
okay,
milk
that,
is mother's milk
yeah.
is retinol.
Okay, yeah, that I do know. All right, that tracks, that makes sense. Or it's starting to come together now.
Right. And so what's one of the most important mechanisms to make copper bioavailable? You got to have
Vitamin,
retinol.
yeah, vitamin A
If you don't have
in its
retinol
active
to
form.
activate the copper pumps, nothing happens. And so we live in a world now where people are afraid of I like to call it unprocessed dairy. Raw sounds so crass, but it's raw dairy is where the action is. The real insult to milk was the homogenization. It was the breaking up of the fat. That's where the problems really started. But lactoferrin, very, very important. Um, and I think one of the better articles that was written during COVID was by Douglas Kell and Etheresia. Uh, I'm blanking on her last name. Um, in any event, Douglas Kell, I think it was 2021, talking about lactoferrin and the whole dynamic of the viral activity during COVID. People, I don't have the title memorized, but it would be a wonderful article for people to familiarize themselves with, because it really gets into the weeds of why lactoferrin is so important and how it interacts with ceruloplasmin. And it's like, it's really, really important, especially in those molecules that we're talking about with macrophages, which are intensely important for our health and well-being. And they are, you made the comment about ferroportin, the iron doorway. You've got to be able to, the macrophage is going to engulf the problem and then digest the problem, but it's got to get rid of the iron. It's got to get out of the macrophage, and that's through a doorway called ferroportin, which requires the ferroxidase enzyme function. And that's not openly taught in practitioner school. There's no awareness of this need to have iron recycling throughout the body, and so there's a lot of confusion. And the the other nuance to lactoferrin is it becomes apo and holo. Did you know that?
No, I didn't. Could you
So
explain that slightly? Yeah.
apo is empty, holo is full. So they actually sell people full lactoferrin that has iron already attached to it, because we're anemic, don't you know? When in fact what we really need is apolilactoferrin, like Mother Nature produces in the cattle, so that we can gobble up the iron that's the cause of all of our problems. It's iron. The concept of aging, everyone's into longevity now. All the really hip scientists are talking about longevity and reversing aging. Well, what is longevity?
Lack of oxidative stress.
Lack of oxidative stress. And what's another way of saying aging? Iron
Uh,
accumulation.
yeah, that makes complete sense.
Yeah,
Yeah,
and
yeah.
that's the work of Denham Harmon from 1956, He was a PhD industrial engineer who studied oxidative stress in the industrial setting. And he was in his 30s. He thought, gee, I wonder if this applies to humans. And so he decides to become a doctor. And he goes to Stanford. So he's not your average chimp, and gets his medical degree. And he creates this whole concept of the free radical theory of aging, which put the world of conventional medicine on its ear. And then 50 years later, he was 40 years old when he wrote the first article. When he was 90 years old, he updated his findings. And it's like, it's the most accepted model of aging on the planet, is Denham Harmon's free radical theory of aging. And we're back to the three-ring circus: iron, oxygen, and copper. We got to manage those three elements. And that's the ultimate goal of the root cause protocol, is that when they are managed, you're going to make more energy, you're going to clear more exhaust, and you're going to have fewer symptoms. That's the whole basis of the argument. And it seems to bear witness in the people who do it on a regular basis.
Morley, that was fantastic. Thank you. I think that's a perfect place to end it as well. You were a terrific guest. I think you've already suggested as much, but where can people find you if they'd like to learn more, maybe specifically about you and your protocol?
Absolutely. People are welcome to buy my book. It's called Cure Your Fatigue. There'll be a second edition coming out in November, so you can certainly enroll for the second edition. You can still buy the first if you want. The website, rcp123.org. We have a community, the RCP community, that meets every other week. Yep, meets every other week, and people get to ask questions, and I'm always there. We have an institute where we train people about these principles. It's a 16-week program, and would welcome people being a part of that. We're in class number 4 of 16 in group number 22. We've trained just under 1,000 people now, coming up on 900 people. Very exciting. And we're on all sorts of social media. And then I'll let people know that they can reach out to me personally at my email address, morleyrobbins@gmail.com. Or for the brazen few that want to call me, it's area code 847-922-8061. And I've never met a question I didn't enjoy, but I appreciate the chance to help people understand these concepts and really value our time together in that regard. So thank you for the dance, if you will.
Definitely. That's very gracious of you. And thank you for your time, Morley. It was an honor and a joy to speak to you.
Thank you.